Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover the Laboratory 24 experimental procedures. So, Laboratory 24, we are looking at the electrocardiogram, also known as the ECG, also known as the EKG, depending on who you're talking to. Kind of just different names for the same thing. And we're also looking at pulses. So let's get into it. Oh, before we get into it, there's two lessons that we're doing for this lab. So Laboratory 24 is the lab, but we're doing BioPack Lesson 5 and BioPack Lesson 7. Remember, BioPack is the name of the computer system that we would use if we were meeting live in class to record data. So there are computers and sensors in this experiment. All right, so a little bit of background first. An ECG provides an electrical view of the heart. So we've all probably seen this printout in the movies. You know, somebody's lying in the hospital bed, they have the monitor right next to the bed, and you hear the boop, and then the line kind of goes up and down, does some squiggles, and boop. Then we're gonna learn what that actually means. So electrocardiogram, or the ECG or the EKG, is an electrical recording of the heart. Because of the electrolytes in our blood and because of the electrical characteristics of our heart, those electrical characteristics are able to be carried through the electrolytes in our blood, through our blood, and they're able to be picked up if we put sensors on our extremities. So we can put a sensor on our right wrist, and left leg, and we're able to see what's happening electrically within the heart. That's what we're doing in this lab. So a little bit of review. A wave is the deflection above or below the baseline. A segment is the baseline between waves, and an interval is a combination of waves and segments. This information is in lecture also, so be ready for it for both lecture exam and lab quiz. So P wave, deflection away from baseline. T wave, a deflection away from baseline. Segment, ST segment, the baseline between waves. An interval, PR interval, it goes from P to R. P to R, a combination of waves and segments. And then we also have this QRX complex right here. In lab 7, we're actually gonna hook up one student to the ECG, and we're also gonna hook, that's what's going on this right side of the screen, and we're also gonna hook up this pulse pressure sensor. So the ECG is going to see what's going on in the heart electrically. The pulse pressure measure is going to measure, so you know how, like, if you take your pressure, your pulse in your neck, if you take your pulse in your wrist area, you feel the artery expand and relax and contract, expand and contract. That's what we're feeling for. When your ventricles push blood into, well, when your left ventricle pushes blood into your aorta, it pumps up the aorta like a balloon. And that pressure, that pump up of blood, a wave of pressure goes through your arterial tree. And that's what we're measuring out here on the end of our fellow student's finger. So that pressure wave starts, it's generated by the left ventricle, it goes through the aorta, through our arterial tree, all the way down our arm, and it's picked up here. So that's what the pressure monitor is recording. We're going to attach the electrodes and pressure monitor. We're going to calibrate, so there's always a calibration phase with the BioPack. And then we're going to have that student sit with their hand on the table, and we're going to measure both recordings for 10 seconds. Or they're going to have them drop their arm below their waist, measure those recordings for wait for a minute and then measure for 10 seconds. Or they're going to have them hold their hand above their head, wait 1 minute, and record for 10 seconds their ECG reading and their pulse pressure reading. How do you think the pressure that is felt, the pulse pressure, how do you think that's going to change with hand position? Do you think it's going to change at all with hand position? How do you think the electrical characteristics of the heart are going to change with different hand positions? Is it going to change at all? So after we do that, we're going to analyze the data. We're going to measure the data for R to R interval. So interval is how long it takes. How long does it take to do one full cycle, to go from R to R on the ECG, one full cycle? How long is each cycle? And then we're going to extrapolate that out to beats per minute, so heart rate, contractions per minute of the heart. How long does it take? So then we're also going to measure the time from one pulse to the next pulse. So the pressure that is felt at the tip of the finger. One pulse to the next pulse. How long does it take? And then we're going to extrapolate that out to a full minute. So how long does it take, or how many pulse pressure waves do we feel within a minute? Are the two related? If so, how? How are they the same? How are they different? Is our heart rate and R to R interval going to change with different hand positions? Is pulse interval and pulse rate going to change? Is pulse amplitude going to change? Is ECG amplitude going to change? What's going to change? Think about it before you watch the discussion video. Perfect. So first off, we start with the hand on the table or at table level, then hand down, then hand up. Perfect. So right here, this is what the data will kind of look like for the pulse pressure wave. We'll see that pulse wave come through, relax back down, go up, come through, come back down. It's just like when you're taking somebody's pulse, you feel that expansion of the artery. That's what it's measuring. All right, so right here's our data table. You can see arm resting on the table, R to R interval, so the interval for pulse, for the ECG, interval for ECG, extrapolate that out to a whole minute for heart rate. The pulse interval, so for how long does it take for in between the pulses, and extrapolate that out for a whole minute. And then we're going to do that for the 3 different arm positions. We're also going to see how QRS amplitude changes in the different positions. And we're also going to see how the pulse amplitude, so how much expansion is there for that pulse wave, how intense is that pulse wave for each arm position. How is QRS amplitude going to change? How is pulse amplitude going to change? All right. For lesson 5, so this is the second part that we're doing. For BioPack lesson 5, it's going to be similar. We're not going to do anything with pulse pressure wave, but we're going to do a deep dive into ECG. So we're going to leave the student, the experimentee, hooked up to the ECG. We're going to leave the electrodes on. We're going to follow directions on the screen for laboratory 5, lesson 5. And we're going to collect the data. So calibration should have already occurred in lesson 7, so we should be good with calibration. We're going to have the student leave their hand on the table, and we're going to measure we're going to create an ECG while they're taking 3 deep, slow breaths. We're going to so when you press the F4 key and the F5 key, it puts a little mark in your data so you know where that began. We're going to mark when they start to take in the deep breath with F4. We're going to start when they mark when they start to exhale with F5. So it's going to put a little mark on your data, like where did they start to inhale deeply and where do they start to exhale deeply. We're then going to unhook them and have them exercise for 60 seconds, enough to make it so that, they're breathing hard. And then we're going to hook them back up and reanalyze without the deep breaths, but just hook them back up and just see how the ECG changed. Is the ECG going to change if a person's inhaling or exhaling? If so, how? Is the ECG going to change before and then after exercise? If so, how? So an ECG can estimate the duration of systole and diastole. So remember, the P wave corresponds with atrial depolarization, which is atrial contraction. Atrial repolarization corresponds with the QRS complex, so that's relaxation for the atria. At the QRS complex also, we have ventricular contraction. Ventricular contraction continues until the T wave. We then have diastole throughout both chambers in this section right here. So atrial systole is P wave to QRS. Ventricular systole is QRS to T wave. How are those values going to change in these different situations? How is the TQ and QT interval going to change? How is the PR interval going to change? Hypothesize. I'll see you in the discussion. If you have any questions, as always, please reach out to me. I'm here.